Okay, 3 weeks to go after this. Finish line's in sight. So reading quiz Monday is 14.2 and 14.3. That will be information we will have covered in class. It'll include things about translation and modification of messenger RNA that we talked about yesterday in class. What do they need to do for
Just please read over the protocol for week 2. It's familiar because it's transformation, and that's about it. Although on Monday, I would like to say some things about how to interpret the gel.
Okay, and we'll talk. We'll do more with the blue-white selection again too to make sure that they come comfortable with that. Okay. So, any questions about anything you went over yesterday?
Surely I wasn't back.
Please remember that these modifications of messenger RNA are unique to eukaryotic genomes and not prokaryotic genomes. Those modifications, particularly the existence of introns, create some interesting problems when you try to genetically transform organisms, because if we take a gene from a jellyfish, for example, like you did, and it's got introns in it, and then we insert that into a prokaryotic cell, and we want that prokaryotic cell to express that gene, it's not going to know how to handle those introns, because it doesn't have the machinery, doesn't have the SNRPs, for example, to splice those things out. So there's some tricks you have to play when you genetically modify organisms with DNA that contains introns. That's a little beyond the scope of this particular course.
How deep into eukaryotes does the presence of introns go? Does it go all the way to unicellular eukaryotes?
I believe so, yeah.
Yeah?
I believe so.
I didn't know that.
So you talked about mutations toward the end of the class period. And all 3 of these particular types of mutations were discussed. The first one is pretty innocuous. It has no effect on the amino acid sequence because the substitution that occurred, here is our wild type sequence right here. The substitution that occurred only involved this particular nucleotide. Changing it from a C to a U. But this still codes for the same short polypeptide chain because of a particular property of the genetic code. What's that property? Redundancy, right? Okay. So this particular modified code word still codes for the same amino acid. So no harm, no foul. We make the exact same polypeptide. If we get this particular type of a change in the sequence of the DNA, and then that's transcribed into mRNA, that's a problem because we now change a coding sequence into a different one, and because the first base is the one that's altered as opposed to the third one. So now we actually have a protein that's produced, but it doesn't make sense. It's got a different sequence than the wild type sequence, the normal sequence. And that can have a variety of effects. I mean, it might not really have any effect. If this particular amino acid doesn't have any real impact on the way that polypeptide chain folds up, you know, your protein is still technically mutated, but it still does the same thing and behaves the same way. But sometimes this one single change can have a big consequence on polypeptide folding and therefore structure. And that's what we'll talk about a little bit more in regard to sickle cell anemia. Yeah, Janet?
Is it only a missense if it's at the beginning, or is it a missense if it changes it completely?
Okay, here, if you change with missense, if you change either the first or second nucleotide, that will change an amino acid. If you change the third one, it may or may not, depending upon the coding. Okay, because sometimes there are only a couple of redundancies, sometimes there are 4 for a particular amino acid. But you're pretty much guaranteed to change the amino acid coded for if you change the first or second nucleotide. Okay? And then this is very serious. This nonsense mutation, we're turning a code word for an amino acid into a stop codon. And so this would then just terminate the synthesis of protein at methionine, and we just wouldn't make the rest. And that would have likely disastrous effects on the function of that particular protein. What do all these have in common in terms of the typical cause of these particular mutations, of these no effect, missense, and nonsense? What's causing them? You're saying something? What do you say, Ken? Okay, a change in the nucleotide, we have a substitution. Why is that happening? And when's that happening? During DNA replication. Okay, we've got a base pairing mistake that occurs, and then that DNA polymerase editing enzyme hasn't picked up that particular mistake. That 1 in a billion ones that get through. Okay? Well, I'd like to concentrate on this missense just a little bit more. The consequences of making a missense protein are really threefold. I'll give you one. There might not be any consequence at all. If that particular amino acid is inconsequential in the way it folds up, it just doesn't matter. Okay? What are the other two possible consequences of this missense mutation on the organism? Is that
helpful?
It could be helpful. It could be a beneficial mutation, or it could be harmful. It could lead to a loss of function. Okay? What sickle cell anemia is very interesting about is that it results in a single nucleotide substitution. It is a missense mutation. It is a spontaneous mutation that occurred as a result of errors in replication. And it did have an effect on hemoglobin molecules. What's interesting about this is that this particular mutation, the effect can be positive in some circumstances, but it can be negative in other circumstances, depending upon whether it's on both chromosomes. Remember, we have homologous pairs of chromosomes for all of our genes, including hemoglobin. And also depending upon the environmental conditions. What is the major environmental condition that affects whether this hemoglobin mutation is helpful or harmful? You saw it in the lab, in your video. Okay. Malaria. The existence of the mosquito, actually, that transmits malaria. Okay? So in areas where this mosquito is endemic, where it tends to live naturally, then there's a much higher probability that people will contract malaria because mosquitoes are the vector for that parasite. So in those particular environments, having this particular mutation can actually be helpful. So here's our mutation. Here's wild-type hemoglobin DNA. And this is the 3-base codon down here that ends up being affected in our particular mutation. If we have a base substitution of T being substituted by A, then this particular codon is altered from GAA to GUA in the message. And the hemoglobin then has glutamic acid in the wild type form, and in the mutant form it has a different amino acid. It has valine. Okay? Now, to know something about these particular amino acids would give you some insight into, might this have some effect on how the hemoglobin folds up? And it actually does, pretty significantly. That's because this tends to be a charged acidic amino acid, and this tends to be more of a nonpolar amino acid. So knowing something about the properties of the amino acids can be important. So we got this situation where we've got the wild type hemoglobin gene, and we've got the mutant hemoglobin gene. I'm going to designate the wild type gene as a capital S and the mutant gene as a lowercase s. Okay? This particular gene, this mutant gene, is called the sickle cell gene. Okay? So if we've got an allelic pair, a pair of alleles governing a particular trait, how many particular different types of genotypes can we have? We can have this one, right? Or we can have this one. Or we can have this one, right? So homozygous dominant, heterozygous, homozygous recessive. Okay? This individual is considered to be quote-unquote normal. This particular individual suffers from a very debilitating disease called sickle cell anemia. And you saw the effects of sickle cell anemia on the little girl in Africa who had sickle cell. And there were all kinds of physiological consequences associated with that. We'll show you a slide in just a second. But this particular disease can be deadly. The individual, the heterozygous individual, is considered to be a carrier for the trait. But more so, in addition to being just a carrier, these individuals are an advantage under certain conditions, the presence of malaria. Because this gives you, being a carrier, gives you a resistance to that particular disease. That particular concept is called heterozygote advantage. And in this particular case, there's a real advantage to being resistant to the disease malaria. So what's going on here? Well, here's our normal red blood cells. Red blood cells tend to form these kind of really nice, simple-looking biconcave discs. They're kind of indented in the middle. And these things move through small spaces like capillaries quite easily. Here is the normal hemoglobin sequence of amino acids. And here is our mutated or sickle cell lowercase s amino acid sequence. One amino acid substitution, but look at the effect that this has on the red blood cells if you have both of these alleles. If you have both of these alleles, a large percentage of your blood cells are just permanently sickled. What effect do you think this might have in terms of ability of those blood cells to move through tiny spaces like capillaries? Going to be as easy as these? No, these are going to get hung up, aren't they? And they can get caught, and they can start to kind of stick to one another. And the capillaries can literally get clogged up if too many of the red blood cells are sickled. And sickled cells clogging up your capillaries can have all kinds of physiological consequences. The body tries to break down these altered blood cells. They tend to become clumped and they clog small blood vessels. They tend to become accumulated in the spleen. The spleen is one of the parts of your body that helps to break down old red blood cells. And these can lead to these particular consequences. Okay, physical weakness, anemia, heart failure, pain, brain damage, kidney failure. Lots of nasty things here. Okay? So this particular condition is deadly. Okay? Well, what's interesting about sickle cell is that if you look at it, the frequency of sickle cell alleles varies significantly depending upon what part of the country you live in this particular case, Africa. So greater frequency of these alleles, more red the color on this map, greater the chance, if you have a greater frequency of those alleles, that individuals will be homozygous recessive. And as a matter of fact, these high frequencies don't really make sense to geneticists because in these particular conditions, these individuals tend to kind of be selected against because they don't survive and reproduce. And so the frequency of sickle cells kind of exceeds expectations many times. They go, what's going on here? I mean, there are just too many of these sickle cell genes. It doesn't make sense. Well, the reason is because this gene tends to be maintained in the populations in the heterozygotes because of the advantage that heterozygotes have. What's your advantage? Your advantage when you're a heterozygote is that your cells will tend to sickle. Bad? Possibly. But typically only if the cells get acidic inside, if the red blood cells get some acid in them. Well, why might red blood cells get acidic? If they become invaded by the malaria parasite, and that parasite metabolizes food and produces acid as a byproduct. And what happens to a sickled cell? Your immune system, white blood cells, recognizes those sickled cells and says, don't belong here. I'm going to get rid of them. So people who have this particular genotype have kind of a defense mechanism built into their genes that basically signals to the immune system red blood cells that have been invaded by parasites, and then the immune system kills them. Okay? These individuals, their cells are constantly in that kind of sickle-celled state. These individuals, typically you only get the sickled cell when they become invaded by the parasite. Okay? So very interesting genetics and very interesting consequences as a result of that particular condition. Okay, all of the mutations we've been talking about so far are the result of errors in replication, base pair substitution. They usually involve only a single nucleotide. There are some mutations, however, that can be caused by other factors, and they're called induced mutations. And these are caused by environmental damage. What kind of things in the environment might damage your chromosomes? Say something, Nothing? Forms of radiation, like ultraviolet radiation or X-rays, for example. Any of those parts of the electromagnetic spectrum, remember that's the energy that comes from the sun, that are fairly high energy can disrupt your DNA. What's something else in the environment besides radiation that could potentially cause damage to your chromosomes? Chemicals, okay? There are some chemical compounds that can damage your DNA as well. We're going to talk about a particular type of mutation that can be the result of an induced mutation called a frameshift mutation. Oh, I didn't mention this before. With ultraviolet radiation, remember we have repair systems to repair those thymine-thymine dimers that ultraviolet radiation can cause. We have excision repair enzymes. There are some individuals who have defective repair enzymes. And so if their skin gets damaged by UV radiation, they can't repair that damage. And therefore they are much more prone to skin cancer. That particular condition is called xeroderma pigmentosa. So it is a highly elevated risk of skin cancer because of defective DNA repair enzymes. Individuals that suffer from this particular condition have to take real certain steps to try to expose or try to avoid exposure to sunlight. So if they go outside, they've got to completely cover up with sunscreen. You've got to wear protective clothing, hats, long sleeve shirts and things like that. And so the lack of those particular repair enzymes can be really consequential in terms of the development of this particular disease. Chemical compounds, what do they do? How do they cause induced mutations? Well, typically what they do is they will cause either a base pair addition or deletion from DNA. So they actually will either add a nucleotide to a DNA sequence or chop one out. How can a chemical add a nucleotide? Well, a lot of these chemicals are called DNA nucleotide analogs. And these are molecules that kind of look like and they'll be interpreted as another nucleotide being in the DNA. Other chemicals can cause a nucleotide to actually be cut out of the DNA. Both these additions and deletions can cause this induced mutation called a frame shift mutation. We're going to see in a second that the worst kind of addition deletion involves either one or two nucleotides at the beginning of a gene. And we'll show you why that's the case in just a second. We're going to concentrate on induced mutations, one caused by the environment, in this case chemicals, that cause nucleotides to be either added or taken away from the DNA, resulting in this frame shift mutation. So what's going on here? Okay, so here's our normal messenger RNA coded for by a normal sequence of DNA. What if, and these are the amino acids that are coded for by that particular sequence of nucleotides in the message. So methionine, lysine, phenylalanine, glycine. So what if a chemical mimics a nucleotide and becomes inserted into the DNA, and then that's copied into a messenger RNA? So effectively, we've had another U added to this message. Remember how messages are read in groups of 3 nucleotides at a time. So this stands for methionine, this stands for lysine, etc. So now, in this messenger RNA with the added nucleotide, the first codon isn't affected. But when we have that addition now, that shifts this reading frame of nucleotides over by 1. So now the cell is looking at this, and it should be saying AAG lysine, but because we've added a U in here, it goes UAA, and in that particular case, UAA is a stop. So in this particular case, the frame shift, the addition of the nucleotide, causes immediate nonsense. Protein stopped. Over here, we've had a deletion of a nucleotide. Okay, it's supposed to be UUU. Some chemical has caused the nucleotide to be cut out of the DNA, and therefore the messenger RNA is missing a nucleotide. So from the point of deletion on, we get a frame shift. And all those amino acids after the frame shift are affected. So the first 2 were okay, but then the last 2, different amino acids. So deleting a nucleotide shifts the reading frame and all of those particular codons from that particular point on. Why is an addition or deletion at the beginning of the message more consequential? Because then everything pretty much is going to be affected. If the addition or deletion is at the very end of the message, then only a few amino acids are going to be affected. Why is an addition of 1 or 2 nucleotides worse than 3? Well, down here, if we have a deletion of 3 nucleotides, then we're missing this whole codon, and we're missing 1 amino acid. But there's no frame shift occurring because we've deleted 3 codons. Okay? So we get a methionine, phenylalanine, glycine. We should get methionine, lysine, phenylalanine, glycine. Okay? So it's important to understand the consequences of frame shift mutation, particularly additions and deletions of 1 or 2 nucleotides, shifting that particular reading frame and affecting everything from that point on till the end of the message. We're going to talk about the molecular basis of cancer after we talk about regulation of gene expression. One of the things that I want to tell you right now is that there is a high degree of correlation between mutation of DNA and resulting cancer. That is typically the first initiating event is 1 or more genes becoming mutated when cells become cancerous. In looking at chemical compounds that cause cancer called carcinogens, 90% of those are also mutagens. Okay, so there's a high degree of correlation between chemical compounds that cause mutation and chemical compounds that cause cancer. This gives us an interesting tool to do some fast and dirty studies about what kind of particular compounds might be carcinogenic. Is that useful knowledge? You bet. Okay, you would want to know, for example, if some company's new food additive was potentially carcinogenic, because you wouldn't want them putting it in your food, right? You don't want to get cancer. But testing compounds for their carcinogenicity to see if they're carcinogens is really a time-intensive, expensive, laborious process, usually involving animal studies, and it just takes a lot of money to screen compounds for being carcinogens. But what if we tested them for being mutagens instead? If they turn out to be a mutagen, there's a high degree of probability it would be a carcinogen as well. And then those animal studies could be done. But if they aren't mutagenic, then the chances are they're probably not carcinogenic. So doing this fast and dirty test on chemical compounds that are suspected carcinogens to see if they are mutagens is a pretty cost-effective way to go, and it's worked pretty well. There's a particular test that's been developed and in use for a long time called the Ames test. And the Ames test screens potential chemicals for being carcinogens by seeing if they're mutagens. What's so special about the Ames test? The test subject are bacteria. So we're not talking about animals and having to grow animals and expose them to chemical compounds. We take bacteria growing on plates and expose them to chemical compounds. And I won't go into how this particular test works, but basically you can tell by looking at how they grow or don't grow on a plate whether they have been mutated. So basically what you do is you just take a bacterial plate and you put a little hole or well in it, and you can put your chemical there, and then you smear bacteria on the plate, like you did last semester with the mouthwash experiment. And then you look to see whether the growth of these particular bacteria are affected or not. If the growth is affected in particular ways, then that means they become mutated. And if they become mutated, then the chemical compound has caused the mutation. And then we can say, uh-oh, we want to follow up now and do other studies on this to see if it is a carcinogen as well. But we'll come back and talk about the molecular basis of cancer more in a while. Okay, so that's everything from chapter 14. So let's hop over now to the next chapter. And talk about the regulation of gene expression. Okay, what does it mean to say a gene is expressed? What happens if a gene is expressed? A protein is made, right? So the information that is encoded into the DNA gets turned into a message. That message becomes translated into a polypeptide chain. So genotype ends up being expressed as a phenotype. Okay? Both prokaryotes and eukaryotes can change the expression of their gene in response to changes in their environment. And we're going to spend a fair amount of time talking about how certain prokaryotes do that particularly. Multicellular eukaryotes can also do this as well. But additionally, regulation or changes in gene expression is also important in multicellular eukaryotes during the process of organismal development, and particularly is responsible for the development of different cell types in a multicellular organism. So by turning on and turning off genes at critical times, An embryo can develop into a fetus can develop into a young baby, and the baby can have differentiated muscle cells and skin cells and liver cells and brain cells. That's all the result of changes in expression of genes in those particular cells. Gets pretty complicated. There are various interactions that are going to be really important in changing the expression of genes and turning them on and turning them off. Proteins can interact with DNA and alter gene expression. RNA can interact with DNA and alter gene expression. And RNA can react with other RNA molecules to alter gene expression. We're going to start out with some simple models that illustrate how prokaryotes can change the expression of their genes. And then we'll just scratch the surface a little bit about regulation of gene expression in eukaryotes, with the kind of idea in mind that this is going to lead up to an understanding of cancer, because cancer is the result of abnormal gene regulation and turning on and turning off genes at the wrong times or the wrong places. So let's take a look at bacteria. Bacteria often respond to environmental change by turning on or turning off the production of certain proteins. Why is it important for bacteria to be able to do that, to turn on and turn off the production of certain proteins? What does making proteins require? A lot of energy. Okay? So natural selection has favored bacteria that can produce only the products that are needed at a particular time and in a particular set of circumstances. You've got a whole bunch of bacteria living in your gut. And you eat different things on different days. And so the composition of foods in your gut is constantly changing, right? Let's say you're a big milk drinker, for example, and then all of a sudden you decide, don't want to drink milk anymore. Dairy products are your primary source of the sugar lactose. Okay? So depending upon the choices you make, drink milk or not drink milk, the lactose concentration of your gut can vary tremendously. Well, lactose is a potential food source for bacteria. And if you're drinking your milk regularly, it makes sense for the bacteria to produce the enzyme that breaks down lactose. What's that enzyme? Beta-galactosidase is the name of the enzyme. Okay? But if you're not consuming dairy products and there's not a lot of lactose in your diet, does it make sense for bacteria to produce that enzyme that breaks down lactose? No. It's a waste of resources. Well, when it comes to enzymes, a cell has got some options in terms of how it can regulate enzymes. One of the things that it can do is it can regulate enzymes through the process of feedback inhibition. Do you remember how feedback inhibition works? The end product of a metabolic pathway can go back and inhibit the activity of that particular enzyme. But we're still making the enzyme, right? We're just shutting the enzyme off temporarily. Another possibility is to regulate the gene and not the enzyme, and either turn on or turn off the production of the enzyme. So there's kind of two different levels at which we can regulate enzymes. Enzyme activity by feedback inhibition, or actually regulate gene expression, turning on or turning off the gene. Which one of these saves more energy? Obviously this one does, not producing the protein in the first place. Which one of these allows faster response to changing conditions? Regulating enzyme activity, because the enzyme's already there. It's just a matter of whether the enzyme is in an active state or an inactive state. So both strategies make sense. Both really have different functions. Okay? So, here's regulation of enzyme activity. Here's some metabolic sequence where some precursor molecule is converted into an end product like the amino acid tryptophan. This particular pathway involves several different enzymes. Tryptophan, the end product of this particular pathway, can feed back and inhibit this first enzyme in the reaction. So if there's a lot of tryptophan present, it doesn't waste the precursor making more tryptophan. It can use this precursor to make something else if it needs to. Okay? Another way you can regulate this, though, is to regulate the expression of genes. Regulate whether these enzymes are produced or not just whether they're active. So, for example, tryptophan can feed back and affect the expression of this particular gene, and temporarily shut off the production of all of these enzymes. Okay? That's going to save you some resources. But this is going to take longer. This is a slower means of regulation, but it does save you more energy. This is a faster means of regulation, but it doesn't save you the energy needed to make these particular enzymes. We're going to be talking about regulation of enzyme production involving regulation of gene expression. Okay, in the 1960s, long time ago, a couple of French biochemists developed a concept that has become a really important concept in gene regulation. And that particular concept is called the operon model. And they hypothesized that bacteria can regulate expression of their genes, turn them on and turn them off, and that the way they did that is explained by this concept of the operon model. Happened in 1961. The operon consists of several elements, and these are all part of the bacterial's genes. Okay, so these are DNA elements. There are repressor genes, operators, and structural genes. The structural genes actually code for enzymes. Someplace else on that bacterial's DNA is another type of gene called a repressor gene. And that codes for a repressor protein. Now, repressor protein isn't an enzyme. So when we're talking about enzymes, we're talking about these guys right here. Okay? The structural genes code for the particular enzymes, but there's another kind of gene that codes for something called a repressor protein. Adjacent to the structural genes are two important regions. One is called a promoter, and one is called an operator. Okay, so these are a bunch of nucleotides. These are a bunch of nucleotides. These are nucleotides. These are nucleotides. Okay? Do you remember what the role of the promoter is in transcription? That's the place where the RNA polymerase attaches. And if the RNA polymerase attaches and then starts moving down and reading the structural genes, what do? We make a message, and then that message is translated into proteins. that RNA polymerase will move down and transcribe those genes if it attaches to the promoter, if it can get past this region called the operator. The operator represents a potential roadblock. And if there are traffic cones occupying that region right there, it blocks the ability of the RNA polymerase to transcribe those genes. If the traffic cones are taken away, then transcription occurs. Okay. So what are our traffic cones in this particular analogy? These repressor proteins. And the repressor proteins can occur in two forms, or two conformations, or two shapes. An active state and an inactive state. If the repressor is in the active state, then it binds to this operator and blocks transcription. If it's in the inactive state, can't bind. So what's going to determine whether this particular repressor protein is active or inactive? Whether it's able to block or not block? Well, there's going to be a binding site on that repressor protein. And that binding site is a place where some other molecule can bind to that repressor protein. And depending upon the nature of the repressor protein, if the other molecule is bound in some repressor proteins, that makes them active. In other repressor proteins, it makes it inactive. So, protein interactions are going to be important here. But ultimately, what determines whether these genes are transcribed or not will be protein-DNA interactions. The protein actually interacting with the gene and binding to the gene. So, what's an operon? An operon is a group of functionally related genes that can be coordinately together controlled by a single on-off switch. The regulatory switch is this region called the operator. And it's positioned within this promoter that the RNA polymerase occupies. And so actually, this whole thing is a promoter. And the operator is a region within the promoter. This stretch of DNA, consisting of the promoter and its operator and the structural genes, make up this unit called the operon. So technically, the repressor gene is not part of the operon, but it does regulate this particular operon. Now, what's the advantage of this? Well, a couple of advantages. Number one, it gives you a way to turn on and turn off genes depending upon needs of cells. But also, it allows all of these structural genes to be controlled as one unit. We don't have to have individual on-off switches for every structural gene. So go back and look at this metabolic sequence here for a second. Okay? So for example, if we're going to synthesize tryptophan, and we want to be able to turn on and turn off the synthesis of tryptophan by turning on and turning off the genes that code for these enzymes. If we don't need enzyme 1, do we need enzyme 3? Do we need enzyme 5? No. We either need all of these enzymes to make tryptophan or none of them. So if I want to regulate the expression of all of these genes, do I have to have 5 different control elements? No. All 5 structural genes are together, and they're regulated by the same operon, the same on-off switch as a group. So we either make all of them or we make none of them. And that's just a much more simple and efficient regulatory system than having a separate on-off switch for each of those particular genes. Now, there are a couple of different types of operons. We're going to talk about both of them. We're going to talk about a type of operon called an inducible operon, and we're going to talk about another type of operon called a repressible operon. The example we're going to use of the inducible operon is a type of operon called the lac operon. Inducible operons usually involve enzymes that break things down. Lactose being broken down into glucose and galactose. The trip operon is going to be our example of a repressible. Repressible enzymes usually involve the synthesis of some molecule, not the breakdown. In this particular case, the synthesis of the amino acid tryptophan. So what we're going to do is we're going to first look at repressible operons, which is kind of what we've been talking about already with the tryptophan synthesis. See how that works, and then we'll apply what we've learned about trip operons to inducible operons, the lac operon. This is not inherently easy stuff to understand, but once you wrap your head around it, it makes perfect sense. So we're going to spend some more time digging through this on Tuesday. But one of the things I really want to urge you is to try to understand how this works as opposed to just memorizing a bunch of terms. And there are some diagrams that are in your book, that are in the PowerPoint slides, that really very nicely illustrate these processes. And I would strongly urge you, if you're not in the habit of bringing PowerPoint slides, to at least print off those and bring those to class, because if you're sitting here trying to write down all the stuff that are on those PowerPoint slides, you're going to be really concentrating on writing it down and not listening, and you're just not going to make the connections. Okay? So are there any pre-lab quiz next week or anything? Okay, so just reading quiz next week.